So in that particular class, we will going to talk about the gram staining, acid fast staining, and different aspects of the microbiology. Gram staining is a process by which we can easily identify whether the selected bacteria is gram negative or gram positive. Okay, the basic concept regarding the gram positive and gram negative bacteria that I have discussed in the first class of the DSE. I hope you all can remember that particular concept. So here in that particular topic, we need to talk about the basic procedure of the gram staining, right? Now, the most widely used staining procedure in the microbiology is the gram stain discovered by the Danish scientist and physician, that is Hans Christian Joachim Gram. Okay, in the year of 1884, gram staining is a differential staining technique that differentiated bacteria into two groups, as I told you earlier, gram positive and gram negative. The gram stain procedure distinguishes between the gram positive and gram negative by coloring these cells as red or as violet. Okay. Now the gram positive bacteria stain violet. Why? Because due to the presence of the thick layer of peptidoglycan in the cell wall, which retain the crystal violet, and these cells are stained with the crystal violet color, and that will be retained within them. Alternatively, the gram-negative bacteria stain red, okay, which is attributed to a thinner peptidoglycan wall, as because the peptidoglycan wall is thin in case of the gram-negative, so they can't retain the crystal violet, and crystal violet will be washed out, and in place of that, another color, the counter stain that will be retained. Okay, there is safranin that will be retained, uh, and because of which the color of the safranin, that is red, that will be the color of the gram negative bacteria. So basically the peptidoglycan thickness, the thickness of the peptidoglycan layer will be the contributing factor for the violet color and the red coloration of the gram positive and gram negative bacteria. Now, what are the different reagents that we want? These are primary stain, that is crystal violet, mordant, that is grams iodine. Mordant are the basic, chemicals which will help to fix the stain with the entities or the molecules which are present within the bacteria. Next, there is decolorizer. Decolorizer is ethyl alcohol. By means of the application of the ethyl alcohol, we can remove the stain. If the species is gram positive, so because of the presence of the thick peptidoglycan layer, the crystal violet will be retained within the structure, and ethyl alcohol will be unable to remove them. But if the layer of the peptidoglycan is thin, so after the application of the ethyl alcohol is able to remove the crystal violet from the cell wall of the gram negative bacteria, and thereafter we have to apply the secondary stain, that is safranin. And as because crystal violet is retained within the cell wall of the gram positive bacteria, so safranin cannot be retained in that particular place because that place is already occupied by the crystal violet, and they will appear violet. In case of the gram negative bacteria, as because uh, of the thick peptidoglycan layer, after the application of ethyl alcohol, the crystal violet is removed. So whenever we apply the secondary stain, that is safranin, so safranin will be retained in the structure, and we can observe the gram negative bacteria as pink, okay, or as red. Okay, basically that is not the actual red color, that is pinkish red, we can say. Now, what is the basic procedure one by one if we discuss? Step 1, preparation of the microscopic slide. Grease or oil-free slides are essential for the preparation of the microbial smear. Grease or oil on the slide is removed by washing the slides with soap and water. Clean the slide with alcohol and dry it for the use. Preparation of the smear. Smear means bacterial smear, as we came to know about the blood smear. Like this, from the culture media, we have to collect the bacterial species of the strain by means of an inoculation ware. Okay, inoculation ware, by which we have to pick the bacterial smear, and thereafter we need to draw smear over the slide, right? With a sterile cooled inoculating loop, the culture is taken on the slide and spread it over the slide. It is very important to prevent the preparing thick, dense smears which contain an excess of the bacterial sample, and that we should avoid because thick smear can restrict the passage of the light whenever we want to visualize it within the light microscope. A thick smear diminishes the amount of light that can pass through. As I told you, smears typically require only a small amount of bacterial culture because we just need to identify whether it is gram positive or gram negative. Sometimes mixed culture may be present. So within the same culture, we can have the gram positive as well as the gram negative. So if we pick a very small sample, so within that small sample, this kind of microscopic entities are very easily identified. So we never need to draw a very big or spreaded or thick smear. We don't need. Heat fixing after preparation of the smear, allow the smear to air dry. After the smear has air dried, hold the slide at one end and pass the entire slide through the flame of a Bunsen burner 2 or 3 times with the smear side up. And heat fixing kills the bacteria in the smear, and that will decrease the chance of the infectivity of the researcher or the student who execute that particular practical. Okay, take care to prevent overheating the slide because protein in the specimen can coagulate and causing cellular morphology to be distorted. Okay, so excess heating that should be avoided. Now the protocol: place slide with heat fixed mirror on staining tray, addition of crystal violet, and let stand for 1 minute. Thereafter, rinse with the distilled water, addition of the grams iodine, and let stand for another 1 minute. That will be act as a mordant, as I told you earlier. Next, what you need to do: rinse with distilled water. The smear will appear as a purple circle on the slide. Decolorize using 95% ethyl alcohol. Tilt the slide slightly and apply the alcohol drop by drop for 5 to 10 seconds until the alcohol runs almost clear. Immediately rinse with water, addition of the safranin, secondary stain to counter stain, and let stand for another 45 seconds. Thereafter rinse with the distilled water, dry the slide with the blotting paper, or in the air you can also dry the slide, and thereafter you have to view the smear using the light microscope. Okay. So as I told you earlier, that is mentioned over here again, if the bacteria is gram positive, it will be remain the primary stain, crystal violet, and not take the secondary stain, causing it to look violet or purple under the microscope. And if the bacteria is gram negative, so it will lose the primary stain and take the secondary stain, causing it to appear red when viewed under the microscope. Okay, now if we move to the acid fast staining, acid fast staining are that kind of staining procedure that will be applied for another segment or another type of microorganisms which are irresponsive in case or in terms of the gram negative staining, gram staining, right? So basically, the Zell-Nielsen stain, also known as the acid-fast stain, was first described by the two German doctors, Franz Zell and the Friedrich Nielsen. It is a special bacteriological stain used to identify acid-fast organisms, mainly the Mycobacteria, or we can say Mycobacterium tuberculosis, which are responsible for the development of the TB. Okay. Acid-fast organisms like the Mycobacterium contain large amount of lipid substances within their cell wall called mycolic acids. Okay, these acids resist staining by the ordinary methods such as the gram stain. The reagent used are Zell-Nielsen carbol-fuchsin, acid alcohol, and methylene blue. Okay, what are the main components? Zell-Nielsen carbol-fuchsin, acid alcohol, and methylene blue. Acid fast bacilli will be bright red after staining. Procedure: drop suspension of culture onto the slide, air dry slide for 10 minutes, then heat fix slide for another 10 minutes, flood the slide with the carbol-fuchsin. Okay, thereafter you have to add plenty amount of carbol-fuchsin over it. Hold the flame under the slide until the steam appears, but do not allow it to boil. Okay, and after the application of the carbol-fuchsin, you have to place the slide over the flame, and thereafter evaporation of the carbon-fuchsin will be happen, and that will create the steam. Okay, but we should not allow the boiling of that acid fuchsin. Now that allow hot slide to stand for 3 to 5 minutes. Rinse with the tap water after it. Okay. Flood slide with 3% hydrochloric acid in isopropyl alcohol. Okay, there is also called the secondary alcohol. Allow to stand for 1 minute. Rinse with the tap water. Flood the slide with the methylene blue thereafter, and allow to sit for another 1 minute. And thereafter rinse with the tap water. And thereafter you have to dry the slide, that may be in the air or you may use the blotting paper. And thereafter you have to observe the observe the slide under the oil immersion light microscope. Oil immersion means we just need to put a drop of oil basically. Okay, and that is olive oil. And after that you just need to attach the objective lens with that oil, and that will create a continuous structure of oil. And via the oil we have to see the entity of the bacteria, then that will reduce the refractive index. Okay, and that means here sorry, the light will be refracted less. Okay, as because that is a very small entity, so as much as refraction will be decreased, we can visualize more, right? Initially, carbon function stains every cells when they are distained with the acid alcohol. Only non-acid fast bacteria get distained since they don't have a thick waxy lipid layer like acid fast bacteria. When counter stain is applied, non-acid fast bacteria pick it up and become blue when viewed under the microscope. And red appeared entities are basically the acid fast positive, bacteria. Obviously, acid-fast bacteria retain the carbon function, so they have this weight. This kind of, observation or entity that you can observe under the microscope. Now, Mycobacterium or acid-fast bacteria. Mycobacterium is the genus of the actinobacteria. The genus include pathogen known to cause the serious disease in mammals, including the tuberculosis. There is the Mycobacterium tuberculosis or leprosy. Okay, there is Mycobacterium leprae. Okay. Now there is a diagrammatic representation of this kind of mycobacteria.